Perfect. So thank you so much. Bear with me as I move into presentation mode. And give me one more second. Let's see, I should be okay. Can you just confirm that you can see and hear me okay, please?
Yes, we can.
Perfect. Thank you so much.
Thank you.
Well, I think Rana and Maha could not have set this up and teed this off any better. So I'm going to jump right into it, into the interest of time. These are my forward-looking statements and disclaimer. I won't read them, but here they are. I do, because Moderna's COVID-19 vaccine has been authorized, but is not licensed or approved, bear with me, I do have to read this. The Moderna COVID-19 vaccine has not been authorized, has not been approved or licensed by the FDA, but has been authorized for emergency use by FDA under an emergency use authorization, EUA, to prevent COVID-19 for use in individuals 18 years of age and older. There is no FDA-approved vaccine to prevent COVID-19. The EUA for Moderna COVID-19 vaccine is in effect for the duration of the COVID-19 EUA declaration justifying emergency use of the product, unless the declaration is terminated or the authorization is revoked sooner. For more information on the authorized use of the Moderna COVID-19 vaccine and mandatory requirements of the EUA, please see the fact sheet for healthcare providers administering the vaccine and the full EUA fact sheet available at the website you see here. So now, having said that, I want to give a few slides just to kind of lay the
Dr. Carla, apologies for interrupting. If you don't mind, just clicking on the display settings to just change the display, because we can see the presenter view as the two slides, this one and the one coming up next. So there's display setting at the top of your screen.
I see it.
On the left-hand side.
I see it. There it is. There it is. How's that?
No. Yeah. Okay. That's great. Perfect. Thank you.
There we go. Thank you.
Apologies for the interruption.
No problem. Thank you. So again, just to kind of lay the background or the groundwork for the next 20 minutes or so, I wanted to talk a bit about how Moderna is and continues to harness big data to respond to the COVID-19 pandemic. So one of the things that still, to this day gives me pause and awe, I should say, is that Chinese authorities shared the genetic sequence of what was at that point called the novel coronavirus on January 11th, 2020. And 2 days later, the sequence for Moderna's COVID-19 vaccine was finalized. So that was on January 13th, 2020. Our first clinical batch of vaccine was then sent on February 7th to NIH for use in their phase 1 study. The first phase 1 participant 2020. 3 days from sequence selection to being dosed in the first human being, which I think is just, again, speaks to not just kind of these newer technologies and new platforms to get a hold of that sequence information clearly enable us to in this pandemic. And so just for completion, a brief
Carla, so your voice is getting interrupted. Can you check your microphone, please?
It seems Dr. Carla is having some trouble with the connection.
She's back, I think. Carla, we have better internet in Lebanon.
So yeah, I can see the little triangle indicating that she has low bandwidth. So I think she needs a little bit of time for the bandwidth to pick up again. But at least thankfully, Saha, I think she's back now.
Yeah. Carla, can you hear us? I think where we're getting Carla, while waiting for her, maybe Maha, if she's with us, can answer one of the questions addressed to her. Why do we talk essentially about the 2 or more strains of the SARS-CoV-2, such as the UK and SA strains?
Hello?
I'm happy to. Can you hear me okay? Oh, there she is.
Okay, let's see if Carla is ready. Otherwise, we'll take a couple of questions. Carla, do you need to restart your computer or? Okay, go ahead, Maha.
Okay, so yeah, I think it is a very good question from Zaina around why do we only talk about these 2 strains? As I mentioned before, the reason why is because we have sufficient evidence to know that they are more transmissible from existing data. There are, as I mentioned in my talk, and as you know, there are strains constantly evolving. The organism is constantly changing, accumulating mutations. Which of these are important is sometimes hard to predict. But one thing that's key is that we think about the spike protein mutations as specifically of higher concern than mutations elsewhere in the viral genome, and specifically in certain sites of the spike protein. And so we have ability to model and try to understand where on the structure the mutation is, what type of mutation is it, and that allows us to kind of get an educated guess on its functional role. And so that's been guiding, there's been a lot of surveillance around the spike protein mutations in specific. Let me know if I should stop or I should keep going.
I don't see Carla.
Okay.
I think she had to restart her computer.
Okay.
Meanwhile, another question, how can we relate this rate of mutation, 1 mutation every 2 weeks, with the vaccine effectiveness, especially in countries like Lebanon where sequencing is not available?
Yeah, so I think the key here is, again, mutations are going to happen. For the most part, small, mutations elsewhere in the genome are not going to affect vaccine effectiveness. There has been some concerns around mutations in the spike protein, and specifically these, the UK strain and the South African strain. And, so essentially triaging this information on where the mutation is helps us understand its potential impact on vaccination. But I think what Carla will be talking about, hopefully as soon as she comes back on, because I know for a fact this is what they're working on at Moderna, there is an interest in, there's going to be a need to update the vaccine. We know that that's going to happen because the virus is changing. And we know already that there is going to be there's a lessening of the vaccine response at least for vaccines that only target the spike. Of course, there's a diversity of different vaccine strategies. But there is going to be, it's anticipated by the community that there's going to be a need to update the vaccine based on its evolution and its genetic mutations. So we're going to be probably in a scenario similar to what we do for influenza, where we update the vaccine every year. We don't know yet how often we have to update the vaccine, but we are going to. And in fact, I know Moderna is working on a new version of their vaccine. I know other companies are starting to do that too.
Yeah, thank you, Maha, very much. I think Carla is back. Thank you, Maha. Carla, please go ahead.
I am. And so what I'm going to do to save bandwidth, if you don't mind, I'll share and then I'll stop my video. I think I'm suffering from everybody being at home and online, all my daughter included. So everybody is consuming bandwidth right now. Let's see. It seems like I need to be made the presenter again, if that's possible.
Sure, our technical team is on it.
Or if somebody on that side has my slides, they could also bring them up, whatever is easiest at this point.
I have your slides. Lena, would it be easier if I share from my screen?
Once Hani needs to make a presenter. So Carla is now presenter. So Carla, I think you should be able to share your slides.
So it's not giving me, let's see, the opportunity to. It is now. It just came through.
Okay.
Let's do it. Come on, we're very flexible, right? We can do this. I am going to go back into. Okay, bear with me. We're going to do this, and we're going to do this. Da, 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 da. Okay. And then in the meantime, let's roll. So I think in the interest of time, we have all learned a lot about COVID-19 vaccines and mRNA vaccines in the past year or so. So this is a cartoon that depicts how it actually works. So how we get from an mRNA transcript encoded in a delivery system into cells to express the SARS-CoV-2 spike protein to which then the vaccine elicits an immune response and protects against COVID-19. I'm going to say just a few words about our phase 3 efficacy trial because it lays the groundwork for what we're doing in terms of our large-scale effectiveness study in the US. So many of you may have heard of this, our COVE study. We're now launching into, or have launched, COVE Teen, there'll be COVE Kid, but COVE study was a one-to-one randomized, stratified, observer-blind, placebo-controlled safety immunogenicity study. The primary objectives were to show the efficacy, safety, and reactogenicity of 2 doses of 1273 to prevent COVID-19 disease. The study was conducted among individuals 18 years of age or older, and there was approximately 30,000 adults enrolled in this phase 3 study. So approximately 15,000 individuals who received the Moderna COVID-19 vaccine and approximately 15,000 that received placebo. And here you can see the efficacy results from that study. But I think the thing that I want to say about the efficacy trial, because we were in the middle of a public health crisis, so this trial was intended to follow individuals for 2 years after the last dose, for both long-term efficacy and long-term safety. But because of the public health pandemic and because of the crisis that we're in, Moderna and other companies have taken the decision to offer active vaccines. So 1273, you'll hear me say that, that's Moderna's COVID-19 vaccine. So to offer the active COVID-19 vaccine to placebo recipients, because we felt like if we have shown, we've demonstrated efficacy, at our first interim analysis look, and we also have some, safety information that we really could not withhold, ethically speaking, the vaccine from our placebo recipients. So basically what's happened to date is that almost all of our placebo recipients in this efficacy trial have now received either their first or their second, or are on their second dose of COVID-19 vaccine. And so what we are doing is using what's now predominantly a vaccinated cohort of almost 30,000 individuals, and we are still following them over time for safety monitoring. But that brings me to one of the things that we, very quickly realized that we were going to need to do at Moderna, because we knew once we saw that we had hit our first interim analysis for efficacy, we said, okay, that's fantastic, and that's fantastic, news for the world. But how are we going to know what the long-term, efficacy of this is? Because we're going to essentially roll over all of those placebo recipients. It all becomes a single arm trial with everybody vaccinated. There goes our comparator group. So basically what we are doing is we've set up a prospective cohort. So we're going to be doing a real world long-term effectiveness study of our vaccine with a large integrated healthcare system in California, so here on the West Coast. So, and two of the main objectives of this long-term effectiveness study are to look at patient populations that we were not able to capture in clinical trials. And I'll go into a little bit of depth in the following slide. Second is also to evaluate the durability of protection. So like, as I said, because it's almost, I hate to use the word truncated, but in a way we stopped, right? We stopped our efficacy analysis with that phase 3 trial. The vaccinated individuals continue to be followed up for safety, but we do want to understand and further characterize, the benefit risk profile of our vaccine, which includes understanding for how long are people protected. So this long-term effectiveness study that we're conducting, so it'll capture, so it's under the EUA, so the guidelines are under the EUA, the emergency use authorization from FDA. It'll include individuals 18 years of age or older who have received at least one, who have at least 1 year of membership in the Kaiser Permanente Southern California integrated healthcare system. So this is a large healthcare system in Southern California where you can get, I think of it as kind of like one-stop shopping, right? You have access to outpatient records, inpatient records, immunizations, lab results, and so on. And all of this is linked, and the researchers have at KPSC, Kaiser Southern, have access to the EHR data directly. And they have really good longitudinal follow-up of individuals over time. So again, it's a prospective cohort study. So again, you can think about, for those who may not know, cohort, case control, different types of study designs within epidemiology. We're basically taking two big groups of people. Some will get the vaccine, some will not, and then we follow them forward over time, and we compare the incidence of COVID disease in those two groups. Now, again, our goal is to follow patients for up to 3 years, getting at that question about durability of protection of the Moderna COVID-19 vaccine. And because it is being rolled out in the United States, we anticipate by the end of this calendar year, we should have somewhere between 500,000 to up to 1 million individuals who have received 2 doses of Moderna's COVID-19 vaccine within the Kaiser integrated healthcare system. It's a range right now because of all things, when we're looking at in the real world setting, this is going to depend on uptake and also timing with regard to vaccine availability. But based on current projections within the Kaiser system, that's about the number of vaccinated individuals that we're looking to have by the end of this year. And so again, to enable some of these analyses with this large number of vaccinated individuals that we're going to follow over time, we're going to look at the real world effectiveness, right? So different from efficacy. So effectiveness used to be called like field efficacy. So like in the real world, let's pressure test it, right? When a vaccine is timed, temperature abused, when it's perhaps there's an administration error, what have you. It's administered to patients that were not in the clinical trials. What does the effectiveness estimates really look like? So again, the primary objectives of the study are to look at effectiveness of 2 doses against COVID, any COVID disease, and then in particular severe COVID. And by severe COVID, I mean hospitalizations or mortality. And just to give you a flavor of some of the patient populations that this very large cohort with access to, like I said, EHRs, lab data, and so on, enables us to look at is, we want to see effectiveness amongst patients such as those with chronic diseases, so COPD, diabetes, those who are immune compromised. So for example, folks who have cancer, transplant recipients, those who are on immunosuppressive medication. And we'll, of course, always, take a look again against, what is effectiveness of this vaccine over time by age, by sex, and by racial and ethnic groups. And a few more individuals or individual patient populations that we want to explore is to look at, the effectiveness of this vaccine amongst individuals with autoimmune conditions, some of the most common including rheumatoid arthritis, individuals who are frail, pregnant women, individuals who have a history of COVID-19 diagnosis, and then concomitant administration, or sometimes people will say co-ad or co-administration of Moderna with another vaccine. This is, I've marked this a secondary objective as if feasible. Right now, US ACIP does not recommend concomitant administration of COVID-19 vaccines with other vaccines. First, we do know that it's going to happen in the real world. These things always kind of happen, particularly with influenza vaccine. And I think second, ourselves, and I think perhaps other sponsors are also setting up clinical trials to at least see safety and immunogenicity when COVID vaccine is concomitantly administered with other vaccines such as flu. So again, this objective is here for now, just to capture what happen, and also because we do think that clinical practice may change over the time frame of this study. And again, coming back to some of the main question, durability of protection, right? We again want to look at how long is protection provided, 2 doses of Moderna COVID-19 vaccine. So for this study, we're actually going to take interim looks or early looks, if you will. So for the first year, after people have received their 2 doses, we will take a look at our effectiveness estimates every 3 months. In the second year of follow-up, it'll be every 6 months. And then by the 3rd year, we're going to have kind of like the cumulative look of what happened over time. What's also very important for implementation and also for public health practice is 1 dose effectiveness. And we get this question understandably all the time. So the Moderna's COVID-19 vaccine was studied as a 2 dose, so a prime and a boost. We don't know what our 1 dose effectiveness is. So we do know in the real world, it is going to happen. People are going to get their first dose. They're not going to come back for their second dose. So we do have 1 dose effectiveness as a secondary objective in this study. And I just want to make one final point. So within this, large integrated healthcare system, we do have the ability to develop and validate and use things like natural language processing or NLP algorithms to try to apply them to particularly the EHRs and free text within the EHRs when trying to look at some outcomes that are, for which a clinical case definition is particularly difficult and, hasn't been agreed upon, for example, by the scientific community. I'm thinking here really of enhanced respiratory disease, but it can also be applied to things like MIS A and MIS C. The other thing I wanted to say about this large cohort study, we did this intentionally at Moderna because as of now, we do not have any safety signals, but what we wanted is to be sure that if a safety signal was to arise, we have a large cohort of individuals that we know received 2 doses of our vaccine and that we could quickly implement or nest a targeted safety study within this cohort if we needed to. And then just for the last couple of minutes, I think I just realized that Maha and I are probably sitting on some very similar websites every day, as I saw also her graph from the Next Strain website, which again is it's really a fantastic resource. But and I won't spend too much time because I think everybody understands what we mean when we're talking about variants of concerns, and that there are SARS-CoV-2 lineages that you can see here, right? We understand now that viruses exist in a population, right? That it's not just a virus X and that's it. That's the only form, if you will, of it. So this is the global distribution of the SARS-CoV-2 lineages that have been uploaded to GSAID as of the end of last week. And again, as Maha very nicely kind of primed us, a lot of this is built off of what is very strong sequencing efforts that are done in certain parts of the world, right? So particularly in the UK, I would say the US is coming online, perhaps more slowly than we had desired, but is coming online with these efforts, at least at a federal level. But there definitely are some regions of the world where I think, you know, this capability, now that we do have these tools and we do have these open source platforms, perhaps efforts could be, you know, put in place, you know, to kind of build up that information or that ability to get that information to these types of platforms and shared. And again, just when, you know, you're looking, you know, on the right-hand side as a, you know, the global distribution of where the variant known as B117 that was first identified in the UK, and where it's now been reported as of, I believe this is March 2nd. And again, I won't spend too much time on it, but you can see how, as Maha mentioned, that there are, you know, as you know, mutations will arise just as a kind of a part of what viruses naturally do. Some of these will confer no advantage. Some of these will confer a fitness advantage, either being able to transmit more effectively or replicate more effectively once they infect cells. So you can kind of, I think one of the most dramatic, also, you know, graphical presentations of this is where we first, you know, detected this substitution of a D to a G at position 614 in the SARS-CoV-2 virus. And we could see that it conferred an ability to transmit more easily. And I think just, I have to say, you know, in terms of, I'll say a little bit about it, and then I'm happy to take more questions, you know, later about what Moderna is doing, you know, on that front with regards to, okay, you know, we have authorizations in many parts of the world for our 1273 or our current iteration of the COVID-19 vaccine based on the original Wuhan sequence. But as we've all seen, there has been this unprecedented, you know, endeavors in many parts of the world to generate sequence information about SARS-CoV-2 viruses. I think what's happened on our side is that it's actually been this, you know, this massive amount of information that has been very quickly uploaded, right? So I mean, for people internally between myself and my computer scientist colleagues and what have you have been, you know, everything from, you know, watching Twitter every day and other social media platforms where sequences are being discussed and uploaded to, we now have an in-house app that we have developed that every single day downloads what's uploaded to GSAID so that we can not only monitor the occurrence of variants that we're already aware of, but try to look for new variants that might be arising and try to figure out how do we triage those and how do we start to understand those so that we know, you know, what characteristics do we look for example, in a variant that potentially means we're going to move this, you know, into our mRNA platform and potentially, you know, to a new vaccine or a new vaccination, you know, strategy that needs to be rolled out. So at the end of it all, I'll just wrap up by saying that I think from the beginning of, you know, the talk where we went from sequence to, you know, or sequence sharing to confirmed sequence for a vaccine in 2 days. The sharing of SARS-CoV-2 sequence data has enabled rapid development of a pandemic vaccine. We are doing large-scale studies, like many people in the world are, but we are integrating, you know, claims data, EHR data, AI techniques applied to those EHRs to help us as a sponsor understand long-term effectiveness and safety questions that can be answered in these real-world settings with this large amount of data, and that especially that could not have been addressed in clinical trials, either because maybe some outcomes were too rare, or in this specific pandemic context, we took the decision to offer our placebo recipients active vaccine. So again, as many have said, but just for completion, global and real-time sharing of SARS-CoV-2 spike sequences has facilitated sponsors' adaptation of our COVID-19 vaccines if needed. So I mean, this has enabled us to move very quickly to come up with a potential booster strategy. and then also to think about what is our next generation of COVID-19 vaccines potentially going to look like. And genomic epidemiology, now easier, faster than ever before, particularly useful in a pandemic setting. Again, as we've mentioned, though, needing to get that capability to globally in many regions throughout the world so that we can have full representation from around the world of what sequences are being detected, so that we can then enable this shift or adaptation of COVID vaccines if needed. And those are just my thank yous, particularly to my to Kaiser and then to all of my internal colleagues on the computational sciences and clinical informatics and molecular engineering and modeling teams. So that is it for me. Thank you.
Thank you very much, Carla, for this excellent presentation. I think I'm pretty sure everyone needs a 2-3 minutes break just to get some water and go to the bathroom. So let's reconvene at 5:45 for the panel discussion. And during the panel discussion, we'll ask questions that were prepared, but also take questions from the audience. So we'll come back at 5:45. Thank you very much for 3 outstanding presentations.
I saw. Oh, God, I see. Hold on. Nice pictures from the times where we could have real meetings.
It seems like ages ago.
We will have
and it was just last year.
I know. Amazing.
Yeah.
Okay. So, I'm turning Hani, can you